Hello and welcome to this Great Minds event that we're holding virtually. Today you'll hear from a fantastic lineup of speakers who will cover the full breadth of dementia research that is happening at the moment. I'm Ivan Koichev. I lead the Great Minds Register, and I thank you very much for joining.
So what this is, it's the first event that we planned for the participants of our project, and Great Minds is a part of Dementia's Platform UK, which is a major study sponsored by the Medical Research Council, and what it does, it brings together researchers from across the world to work on data which has been collected as part of a variety of cohorts, and Great Minds in particular is our attempt to link researchers with volunteers who are very well characterized, who are very rich in medical data as part of their participation in these cohort studies, because we know that can be key in finding the right people to study at the right stage in order to unlock dementia. For the purposes of this event, we'd like to thank in particular HealthWise Wales, who have been firstly terrific in helping us set up Great Minds, but also in organising this event. The way that the event will run today is that each of our four speakers will have 15 minutes to speak about a different component of the dementia research puzzle, and then we'll have 10 minutes to answer questions on that talk in particular. The way that you can ask your questions would be through the YouTube chat, which is on the right-hand side of the screen. But if you are having trouble accessing that for any reason, you could also use Twitter by using the hashtag GMLive during the presentations. The one thing to note is that there is around a 20-second delay between us speaking and us appearing on YouTube as such. And so what I would suggest is that if you have any question during the presentation, then type it in as immediately so that we can pick it up once we're ready to ask the questions. If the time runs out and we haven't answered any questions, then we will post all the replies on our website that you'll be familiar with, which is greatmindsfordementia.uk in the members section. And after the event is finished, we'll also post the recorded version of it on our Great Minds database. We're also very keen to have your feedback, so we'll send you a short form after the event, and it'll be fantastic. It'll be fantastic if you can let us know. So what we have for you today is, as I said, a fantastic lineup, 4 talks looking at a different angle of dementia research. First up will be Professor Sir Simon Lovestone, who will take us through Alzheimer's disease, if you like, the basics of the disease, but also we'll also talk about the prospects of finding a cure or finding a treatment to prevent the disease or slow its progression. Then we'll hear from Dr. Vanessa Raymond, who will talk about the risk factors for dementia, and specifically what we know about the risks that suffering head injury or drinking excessive alcohol, what we know about these risk factors in relation to dementia. The third speaker will be Professor John O'Brien, who will tell us about the latest research in neuroimaging, so brain scans, and what that can tell us about the dementia process. And finally, we'll hear from Dr. Fateni Setelu, whose research focuses on the experience of looking after somebody with dementia, and she'll talk about what the current research is. So, going over what our project is and why we were set up. So Great Minds is a register of research volunteers who have agreed to be contacted for brain health studies and dementia studies in particular. And the reason why this is now an important part of the fight to develop treatments for dementia is that we've come to realize, and we'll hear more from the speakers about this, is that dementia starts ultimately decades before we have any of the first symptoms of dementia. And so the push now is very much to identify people who are at risk and to study them in detail, and then quite possibly in the future we may be able to offer treatments for dementia even though people don't have any symptoms. Who can take part in Great Minds? This is set up specifically for people who are already part of cohorts. So they are highly characterized as part of their participation in medical studies. At the moment, we have people from HealthWise Wales, as well as from the Airwave cohort. And the value here is that in both studies, people would have attended appointments and they would have done a variety of tests over, in some cases, over many years, which allow us to, if you like, study the trajectory of each individual, and then to be much more informed when we need to invite people into brain health studies. What do you need to do if you want to be part of Great Minds? This is something that you would all be familiar with because you would have joined Great Minds already. But in a nutshell, it involves being invited by your cohort to take part in Great Minds, then logging on to our website, creating a profile, and then we ask you some basic questions that can help us in steering us as to what type of study may be appropriate for you. And then we ask you to do regular memory tests online or through the smartphones. So where are we now? In May 2020, we've had an excellent partnership with these two cohorts, so HealthWise Wales, as well as the Airwave Health Monitoring Study. And on the right, you can see a map of where our fantastic volunteers are located. And we are looking to offer Great Minds to 2 more cohorts by the end of the year. So hopefully we'll build up the number of people who are willing to help with brain studies in the near future. Recruitment to studies obviously at the moment is on hold because practically all research sites are closed down because of coronavirus. But we'd like to use this opportunity to build up the Great Minds database, and so that as soon as we're open, we can start reaching out to people and offer them the studies. That's what I had to say as initial words, and I thank you. I think if we're ready, we can move on to our first speaker, who is Professor Sir Simon Lovestone. Professor Lovestone is a professor of translational neuroscience who I had the pleasure of working with at the University of Oxford. Prior to this, he was at King's College London, the Institute of Psychiatry, and he really is one of the pioneers in dementia research, specifically around defining biomarkers of Alzheimer's disease, and also in looking at very large data sets of routinely collected clinical data, and really trying to tease out any hints about, for example, potential medications that we may have missed, but for example, may have a positive impact on dementia and cognition. Simon has moved on to become the vice president in Janssen Pharmaceutical, and is the head of neurodegeneration. So now he's overseeing a whole program, starting from the laboratory all the way to testing treatments in people. So I'm fantastically excited to have him speak to us. Simon, over to you.
Ivan, and hello everybody. It's really fantastic that Ivan and Heather and others have arranged this meeting, and it's even more fantastic that you're all part of Great Minds. I cannot thank you enough for your participation. It's only through studies or platforms such as Great Minds that will make progress on this disease, and we really do need to make progress. And spoiler alert, I'll tell you the takeaway from this little talk I'm about to give, and that is we are making great progress. And I have never been more optimistic than I am today that we will see a therapy that will slow and perhaps even prevent Alzheimer's disease. I think that is an achievable ambition. I'm speaking to you today in my role as an academic, a professor of translational neuroscience at the University of Oxford, although as Ivan says, I also now lead one of the largest, world's largest pharmaceutical initiatives in Alzheimer's disease at J&J or Janssen, but I'm not speaking for Janssen today, I'm speaking for the University of Oxford. Now I am, as I've just said, very optimistic. I'm really encouraged by the progress that we're making, but it's not enough. And I think, to use a football analogy, which is a game I sorely miss, as I'm sure some of you do too, in order to make progress, we need more shots on goal, we need to shoot earlier in the game, and we need to shoot more accurately. And I'm going to build on that idea of more shots on goal, shooting earlier, and shooting more accurately in this talk. But just a reminder, I know you know this, but honestly, I think it's worth reminding ourselves just how many people are affected by dementia and Alzheimer's disease in particular around the world. It's huge numbers today, and they're going to grow in a way that looks exponential, which is a word that you rarely heard only 3 months ago, but it is really increasing rapidly because of the aging population and the costs. And I apologize for using dollars rather than pounds here, but the costs to the economy are simply enormous. Now, we're living through a strange time. Having a meeting like this where I'm speaking to you through Zoom and not being able to meet you in person afterwards and shake your hands. I'm very sorry about that, but we're all getting used to this. And we're living through very strange times with this appalling disease that's with us. But here's a fact that I think we need to hear more about, and it's a really sobering one. And that fact is that even today, at the very height of COVID-19, there are more people dying every month in the UK of Alzheimer's disease than there are of the virus. And actually, it's even worse than that, because of the people who are dying with the virus, a large number of them sadly have Alzheimer's disease, because COVID-19 is predominantly a disease of the elderly. And as we've heard only too tragically over the last few weeks, it's also a disease that has its most impact in nursing homes. And a very, very large proportion of people in nursing homes or other care situations have Alzheimer's disease. So for all of those reasons, Alzheimer's disease is going to be something that stays with us, taking away large numbers of our loved ones every month, every year, and every decade, and not just once in a lifetime until we do something about it. You're helping with that. And I'm going to try and tell you how we on our side of the scientific bench, as it were, are trying to make progress, and I think really are making progress. So we're making progress with drug discovery, and in drug development and drug discovery, we tend to fit this into 4 parts. So it starts with discovery, where scientists ask what goes wrong in the disease and what might we correct it. That's usually done in the context of a university or something like that. And then you move from that to trying to find a drug that might work. And that's where pharmaceutical companies, small companies, but also including some initiatives like the one I set up at Oxford with my friend and colleague Chaz Bountra in academic context as well, try and find a chemical that might work, a drug that might work. And then you enter clinical studies. And that's where things like great minds and yourselves come to play. The start of those clinical studies, phase 1, asked the question, is the drug safe? And you've heard a lot about this in the last few weeks because of the COVID virus work. Trying to develop a vaccine, you've heard about how those vaccines are going to be tested. And obviously it's a very difficult thing to do. That's phase 1, is the drug safe? Also in the latter half of phase 1, you're trying to find out how much of a drug might we use? And then comes a really critical phase. It's called phase 2. And is the concept right? So in a phase 2 study, what we're trying to do is to say, well, now we have a chemical, it works in the laboratory, we know it's safe, we know how much of it we'll use, but in people, if we do what this drug does it look like we would be having an effect that would be useful for Alzheimer's disease? So it might be an effect on the brain, which we can measure through the kinds of work that John O'Brien does with brain scanning, or it might be measuring what we call a biomarker or test, and I'll come back to that. But it's essentially asking, is the concept of this drug right? And then comes the most important phase. That is phase 3, which is asking the question, which is at the end of the day, the only important question, and that is, does it work? And we measure how a drug works by measuring cognition, this kind of tests you're having in great minds, but also whether people who don't have the disease end up getting the disease or whether they're protected from it. So that's drug development, and it is a fabulously costly business. And you only have to look at these costs and you realize how difficult it is, even for very large companies like the one I work with now, but also countries like the UK that have put a lot of money into research for Alzheimer's disease. You realize how all of that investment isn't anything like enough. The cost of the Alzheimer's disease development pipeline that I've just shown you is in the region of $5 to $7 billion for every single drug. And given that every single drug we've tried up to now has failed, this is a very expensive business. And I suppose if I have one worry, it is that if we don't make progress really soon, countries, companies, philanthropic investors will start to say, maybe we can't do this. Maybe it's just too difficult, too expensive for us. We're not there yet. We are making progress. And I think we're going to see a lot of progress over the next 5 years or so. But this is a very, very expensive business. The first progress that has been made over the now nearly 30 years I've spent working on this disease is we understand a huge amount about the process of Alzheimer's disease. So the patient that you see there, or the woman that you see there, her name was Augusta Dieter, and she was in a way the first person with Alzheimer's disease because she was the patient who was a doctor working in Germany at the turn of the 19th and 20th centuries, and he described this condition that affected her, and a colleague of his, Emil Kraepelin, in fact, who was the founding father of psychiatry, he decided to call this disorder, which was a new disorder to science at the time, Alzheimer's disease. When Augusta Dieter died, Alzheimer got to have a look at her brain. So this was a really new technology. It's sort of the white heat of technology at the time, was being able to do a post-mortem and actually look in the microscope at what was happening in brain tissue. And he saw these two things. Now, what you're looking at there, those grey pictures, they actually come from a patient of mine that I looked after right at the beginning of my career. So this would have been in about 1990 now. And his family, when he died, allowed me to use these pictures, especially for training and education and communication. And I've used them many times, and they've been used by others. And every time I show them, I think about him because he had this disease that Alzheimer saw. The two lesions, it's a technical talk, the two changes in the brain that you see there, on the left-hand side, the round thing, that's a plaque, and it's formed from a protein called amyloid, and that leads on to these little squiggly things that Alzheimer described, and they're called tangles, neurofibrillary tangles, and they destroy brain cells, and it's that gives rise to Alzheimer's disease. Now, when I first started, I went to join the laboratory of my mentor called Brian Anderton, and he and a few other scientists had just discovered the protein that caused those neurofibrillary tangles. It's a protein called tau. And Brian Anderton, set me the task, and others, to say, see if you can find out how this sequence of events occurs. Over the years, the 30 years or since then, hundreds, probably thousands of scientists around the world have contributed to this understanding. We now really understand it very well indeed. We understand many of the genes that affect Alzheimer's disease. They don't cause it, they just increase your risks a bit. We also understand some of the environmental factors like diabetes and head injury that increase your risk. Again, they don't cause it, they just increase your risk. We know that these increase your risk of getting plaques, they increase your risk of getting tangles, and that's what gives you Alzheimer's disease, dementia. Once we understand this, that's the first phase of the discovery program or development program I just showed you. We can now set about finding drugs for this condition. And look, this was in 2019, a large number of drugs. Each one of these, the words don't really matter, but these are the names of the drugs. Each one of these drugs was in a clinical trial last year. The way this diagram goes, in the outer circle, those are the drugs that are in the first phase that I told you about, phase 1. The drugs that are in the middle circle, those are in phase 2. And the drugs that are right in the bullseye, those are phase 3 drugs. And in the third of the pie that's facing upwards, these are drugs that are trying to affect that biology that I've just shown you. And they do so through things that you may have read about actually in relation to COVID, called passive and active immunization. That is giving somebody else's antibody or an antibody you've made, or giving a vaccine. The drugs in the purple third of the pie, these are conventional chemicals or drugs, and the others are more complicated combination type approaches. So many different drugs in development. That is great news. Bad news, every one of those drugs in the middle bullseye that has been tested up until now has failed. Every one of those failures is a $5 to $6 billion failure. Look at the money that is being spent. So are we getting better? Well, I think that we are getting much better at doing this. One of the problems that we've had up until now, and this is my personal opinion now, one of the problems we've had up until now is we haven't had that many shots on goal. So most of those drugs that I've just shown you were just trying to do one thing. They were trying to prevent the formation of the amyloid plaque that I showed you that was described by Alzheimer in Augusta Dieter's brain. So even in 2018, over half of all drugs in late phase were targeting just one disease process. That's not good enough. Given how difficult it is to do this, we need to try and do more different things. So let's have a look at what happened later. What we also know, as well as the plaques and the tangles, we know that in Alzheimer's disease, there are other things that happen. We know that the brain has inflammation, it's inflamed. We know that proteins are clumping together, not just amyloid, not just tau, but many other proteins as well. That suggests there's a general problem in something called proteostasis. These cells that you're looking at there, the neurons, the brain cells, they connect to each other. And the point where they connect or talk to each other is called the synapse. And in Alzheimer's disease, we know those synapses retract. The cells become isolated. Another word we've become unfortunately used to using about ourselves. And when things get isolated, they don't talk to each other. You know, that doesn't work so well, and your thinking becomes disrupted. And we know that synaptic dysfunction is a function of Alzheimer's disease. So why don't we develop not just drugs against amyloid, but perhaps to these other processes in Alzheimer's disease? Well, the answer to that question is we are. Let's look at what happens just 1 year later. In 2018, half of all drugs are being developed against amyloid. In 2019, only a third of all drugs are being developed against amyloid. In 2018, only 4% of drugs are being developed against tau. In 2019, double that, 8% of drugs are being developed against tau, and so on. So just what you're seeing here, a movement, and it started many years ago, but every year we have more and more diverse targets. That reduces the risk. It increases the chance of us finding one that will work. We also, though, need not just to shoot more often, become more diverse in our drug development, but shoot earlier. So this is a general picture of what we think happens in Alzheimer's disease. The solid bar is your thinking ability, your cognitive function as you get older. As people of my age know it, your cognitive function gets slightly worse, not very much worse, I hope, but just slightly worse as you get older. But in Alzheimer's disease, it gets very much worse and people enter dementia. But before that, there's a phase called mild cognitive impairment. But actually, the disease starts even before that in a preclinical phase. If we could do our clinical trials earlier in mild cognitive impairment, or perhaps even in preclinical phase, common sense tells you you're more likely to get a drug that has an effect. Problem is, it's very hard to do those clinical trials. Sometimes those trials are called secondary prevention. So what this means is that if I take a drug before I have symptoms, but when I have the disease in my brain, I just don't know it. And if that drug works completely, then I'll never develop symptoms. And as far as I'm concerned, I've been prevented from getting the disease. And that is increasingly what we're trying to do in our clinical trials. But to do that, we need to know who in great minds is already beginning to get the disease process, maybe 10 years before they have any symptoms at all. And in that, we need to shoot more accurately. We need to be able to diagnose people better and more earlier in the condition. We need to shoot more accurately at the disease. And we now have biomarkers that can detect plaque or amyloid pathology, as well as tangle or tau pathology before people have symptoms. We can do that through a kind of brain scan called PET imaging, or we can do that through a chemical measure in the spinal fluid with a needle into your spine, much like women have, or very similar to women have, for example, an epidural when they're giving birth. Now, both a PET scan and a spinal tap test, they are a bit invasive. They're uncomfortable. They're not painful, but they're uncomfortable to have. You have to go into a hospital to have them done. And increasingly, we've been trying to find a blood test. And one of the biggest advances in the last 18 months or so, I suppose, is it's now very apparent that we will be able to soon have a blood test that is quite good at measuring these pathological processes. Not as good as a brain scan or a lumbar puncture, won't do away without those, but it might be a step in the diagnostic process. But we also want a different kind of test, and that is a test, how can I as a researcher do those proof of concept phase 2 trials? If you remember, I said those are the trials where I don't need to know whether the drug is really working, is it effective? I just give me an idea. Let me do a shorter trial, expose less people to a drug, just to give me an idea, is this likely to work? And for that, we need a progression marker, that is a test that would let me have an idea whether a drug is likely to work or not. And up until now, we haven't had those. Now, I think some of you might be volunteering for the deep and frequent phenotyping study, which is a study that is led by Vanessa Raymond, who we'll hear from later. And what this study is trying to do is to find some measure, might be a brain scan, might be a blood measure, it might be measures of your eyes, a retinal scan, it might be using a phone so that you can measure people's movements. It could be all sorts of different measures, and we're going to try all of these in a short period to see whether any of them act as a progression markers that would allow us to do more, faster, more effective proof of concept phase 2 trials. So I think I'm probably nearly out of time. Let me just say a bit quicker than I'd otherwise intended where I personally think the most optimistic or the most promising therapeutic developments are. And this is on the tau biology. It's what I've been working on for 30 years, along with lots of other people, obviously, around the world. So of course, I'm going to think that's the case. Now here, I can't resist showing you this. In the top of these, you see a maggot. It's a fruit fly maggot. That's a normal maggot. The one on the bottom, you see a maggot that we've tried to make have some of the characteristics of this tau pathology or Alzheimer's disease. And you can see that maggot isn't doing very well. It's not moving very quickly. And if you look on the left-hand side, you can see the clumps, which are a bit like the clumps that we think happen in Alzheimer's disease. Now, in contrast to the amyloid pathologies, we know that tau pathology results in dysfunction of brain cells. We've got lots of evidence from models like this, animal models, as well as from people, that if you target tau, you might have a therapy for Alzheimer's disease. We also know that the more tau pathology you have in your brain, the worse your clinical symptoms. And most interesting of all, we know tau pathology crosses from one brain cell to another. So there's been an explosion of tau-based therapeutics or drugs. There have been small molecules, chemicals that affect tau processes. There have been larger molecules, including these antibodies that you hear a lot about at the moment. And the thing that I find most exciting is the potential to actually vaccinate people against tau pathology. All of these are in development, including in the company that I work for, but also in most other companies and in lots of academic laboratories around the world. I am really hopeful that one of these will work, but if they don't, we also have drugs that are being developed against inflammation, against clumping of other proteins, and drugs that are there to support the potential for synapses to work nicely together. And it might be that we need to combine these approaches. And in cancer, nobody with cancer gets one drug any longer. They get a combination or a cocktail of drugs. I think in the future, that's what Alzheimer's therapy will look like. So a last word, it's not just Alzheimer's disease therapy that we're talking about here. These processes that we're exploring in neurodegenerative diseases also affect multiple sclerosis, Parkinson's disease, frontotemporal dementia, and lots of other diseases. Did I say motor neurone disease? Motor neurone disease as well. And there's an overlap between these conditions. And I'm really hopeful that we'll see progress, not just in Alzheimer's disease, but in many of these other disorders as well. So to finish off, I've tried to show you how we get more shots on goal by diversification of drug development, how we're trying to go earlier in our clinical development to shoot at those goals earlier, and we're trying to make our clinical trials more effective with biomarkers. Is there an end of Alzheimer's disease in sight? Well, you know, this development program that I talked to you about from discovery to phase 3 is typically 10 to 15 years in this field. We're some way along, but currently most of the drugs that I'm most optimistic about are still only in phase 2. So we're looking at probably at least another 5 to 10 years before many of these trials even finish. It's going to take we're in this for the long haul. It's going to take a long time, but I do think an end is in sight, and I think we're making huge progress. And if you're interested in reading a little bit more detail about the talk I've just given, I think this journal article that I wrote earlier this year is available. It's free, and you can download it if it isn't being made available. And I hope it's not too technical. So with that, I want to thank you and apologize, Ivan, for going somewhat over.
Thank you very much, Simon. It's always a pleasure to hear you speak. We have a bit of time for questions. We have a couple that have come in. The first question, Simon, is whether, looking to the future with all of the projections about the numbers who will develop Alzheimer's disease, do we have an idea of what percentage of the population will end up having Alzheimer's disease?
Yes, to some degree, we know that already. It's of people aged over 65, it's about 5% who have active dementia. And of people over the age of 80 to 85, it's over 20% of people who have active dementia. The reality is it's even more than that, because a large number of elderly people have the disease in their brains. And of course, some will pass away through some other condition before anybody ever knows they've got dementia. It's a very high percentage of the population.
Yeah. And Jeff Richards is asking about the link to vascular dementia. So he's saying, you know, for example, the British Heart Foundation is carrying out research on vascular dementia. How good are we with linking in with the vascular research that is ongoing?
It's a really good question, Jeff. I think that we've gone through, it is a bit like a pendulum, really. We've gone through a process where we've become ever more specific in our diagnosis. So people get a diagnosis of Alzheimer's disease or vascular dementia or one of the other forms of dementia. The truth is it's more complicated than that. And the fact, I think, is that most people have a combination of different pathological processes in their brain. And many, many people who have a diagnosis of Alzheimer's disease also have some vascular dementia evidence when they pass away. And that's even more true in the other direction. The vast majority, if not everybody with vascular dementia, has some signs of Alzheimer's as well. So I think our understanding is becoming more sophisticated, and some of the research is becoming really interesting, thinking about how we can combine approaches to both the vascular damage that happens in the brain as well as the Alzheimer's disease damage that happens in the brain. And I think this relates to my last point, that I expect at some point we'll be talking about combination therapies. I hope we will anyway, including for vascular dementia.
Okay. Michael Ben is asking a question that is quite close to the work that we're doing. He's asking, how will people in the pre-symptomatic stage of disease be identified for early intervention, and will this be feasible in a system that's struggling for funding? Where will that fit in the whole healthcare model?
It's a really good question. And Ivan, you might want to, as the lead for Great Minds study, you might want to contribute to this. I think it's really important, Michael, to say at the moment we're only envisaging doing this in the context of research. So we're only considering doing it for people who want to volunteer for clinical studies or in a not completely anonymous studies, because right now there is no clinical reason, there's no health reason for knowing that you have a disease in your brain that is not causing you any symptoms when there are no treatments. In a research context, though, I'm hoping that we will have ever easier to use tests or biomarkers that will be employed in studies like Great Minds, so that people who want to volunteer can volunteer for the right study because their biomarkers show them that they've got the relevant pathology. But we do have to get better in clinical trials. Here's a terrible statistic. In 2 of the biggest trials that have ever been done in early stage Alzheimer's disease, trials that these trials alone cost huge amounts of money, in 2 of these studies, 20% of the people that gave all of their time and took the drug didn't actually have Alzheimer's disease. Honestly, we can't go on like that. You can't run research like that. So doing these preclinical or early diagnostic tests is really useful for research. Right now, not useful in the clinic. Your implication, though, is let me extend your question a bit. Once we have a treatment, how will we do this in a health service that is coming under stress and strain? It's a really important question, and I think we have to not just ask, but we have to demand that this is done when we have treatments that are effective. And I would remind politicians and the people who fund the NHS that Alzheimer's disease costs 1% of the world's GDP every single year. So Alzheimer's disease costs huge amounts of money. The biggest cost is to individuals themselves and to their loved ones, but there's a real financial cost as well. And if we had an effective treatment, we might be able to reduce that financial cost.
And in the interest of time, one last question. I guess it goes in the other direction. So Helen is sort of sharing some personal experiences about bringing her mother to the GP three or four years ago with concerns about cognition, but then not being told that it's severe enough, and now she has dementia. So in a sense, there is this other group of people who are sort of in between, that they're not ill enough, and yet, you know, there's clearly something wrong. What do you say to that group of people? How is the healthcare system looking after them? What is your impression?
Yes, I mean, I share your experience from my own family, and I know how frustrating it is and how angry it can make you, and then how sad it is when you realize years later you were right all along. And I do think it is really important that people get a diagnosis at the point they ask for a diagnosis. I don't think it's sensible to go screening populations, meaning screening people who haven't asked for a diagnosis, but when you or your loved one is worried about memory problems, I think you should go to a doctor and you should get effectively tested. And I think that family doctors, GPs, should be capable of doing good cognitive testing and then refer on for diagnosis in a specialist clinic. Some researchers at John O'Brien's University some years ago showed that half of all the people with actual dementia are not recognized as having that in the community. That was some years ago. My sense is that's changing, and that GPs are now much better advised and educated, and the whole community is more sensitive to Alzheimer's disease. So I think that is getting better. But I absolutely agree with you. When you're worried about somebody with dementia, you should be able to take them to their doctor, and they should be approached sensitively, but a memory test should be done and it should be followed up, and they should go to a specialist clinic. There's no reason today for that not to happen.
Fantastic. Thank you very much, Simon. We have one last question, but I think I'll save it for Vanessa, because it's asking about the link between ECT and risk for dementia. And I think her talk on head injury will be very well suited for that. Thank you very much, Simon. It's been fantastic having you on.
Thank you.
Always a pleasure. So the next speaker will be Vanessa Raymond, who I have the pleasure of working with at the University of Oxford. Vanessa is a senior clinical researcher with a special interest in head injury. She has worked on a study in the US looking at the cognitive outcomes of veterans with head injury, and then has went on to set up clinics in neuropsychiatry of head injury while working at West London. And since joining us, she's been really spearheading efforts in dementia research. So I'm looking forward to Vanessa's talk.
Thank you, Ivan. Just going to share my screen, so hopefully this will work. Okay, so I've been asked to talk today about some potentially modifiable risk factors that could contribute to your risk of developing dementia, including head injury and alcohol. I guess I'm going to echo some of what Simon Lovestone has said about, although this seminar is about successes in dementia research, in terms of what we know about head injury and the risk of subsequent dementia, there's still an awful lot of work to do. Just to give a little bit of context about this, I just wanted to describe where head injury sits in terms of other potential brain injuries. So a lot of, you may hear on the news or in various forms of literature, people talk about brain injury, acquired brain injury, traumatic brain injury. Just to be clear, acquired brain injury is basically any injury to the brain that occurs after birth. And broadly speaking, those types of injuries can be split into ones that are traumatic or not traumatic. Traumatic brain injury is therefore inherently a form of head injury, so one equals the other. But there can be a whole load of other causes of non-traumatic brain injury, so things like infections, strokes, tumours, and also alcohol. In terms of traumatic brain injury, so head injuries, we broadly split that group into whether somebody has what's called a closed brain injury or an open brain injury. And basically that means whether the skull itself has been penetrated as a result of the injury. But you can see from the lists on these on this slide that the causes of both of those types of TBI, as I'll shorten it to, are broadly the same. A few statistics about head injury or TBI. It's been referred to in the past as a silent epidemic. Again, like dementia, it's a massive public health problem. It's estimated in the UK, a TBI is suffered about every 3 minutes, and approximately about 1 million people attend A&E within the UK every year. However, as you can imagine, a lot of people do not go to A&E or to see any doctor if they've had a more mild head injury, so they maybe have just banged their head somewhere at home. If there's not been any severe symptoms, they probably don't go and seek any kind of help, so that a lot of these are probably completely unreported. And of the people who do go along to be seen either by their GP or an A&E, 80% are discharged. So vast numbers of people will present, but will then be discharged with any kind of follow-up. What we do know about brain injury, or traumatic brain injury rather, and its risk for subsequent dementia, is based on the people we actually see presenting to clinical services. So that's inherently going to skew some of our knowledge. Because of this, as I suggested, the vast majority of people who have a head injury have a mild head injury, and this may or may not lead to a concussion. So it's estimated about 3/4 of people who have a head injury, it will purely be a mild head injury. However, even allowing for all of this, head injury is actually the leading cause of death and disability in the over 45. So it's a massive problem, as I said, in terms of public health need. And it's estimated again in the UK, about 500,000 people are living with long-term disabilities. And worryingly, the amount of people at least presenting, or that we know about with head injuries, has increased quite dramatically, probably about 50% over the last decade, particularly in women. And again, I'll come back to that as to what that could mean. But again, like dementia as a whole, it does mean head injuries are a massive cost in a public health perspective, and probably equate to 0.5 of the entire annual global output. So because of this, people like the World Health Organization have identified TBI as a major public health problem with huge unmet needs. What this graph demonstrates is what may cause a head injury or a TBI. And along the bottom of this graph, you can see various ages, and then the percentage of TBIs are represented by the bars on the graph. So what this demonstrates is across the lifespan, the likely cause of somebody getting a TBI varies. So for the very young, it's mainly going to be falls. As you get a little bit older, road traffic accidents, road traffic crashes become an increasing cause of head injury. And as you get older still, so in the over 60s, falls become the major cause of head injury. So not only are we talking about a group that don't always present for medical support or diagnosis or follow-up, we're also talking about a range of causes in this group. And again, that's obviously going to skew whether people seek help or attention after they've had any sort of head injury. What this cartoon tries to demonstrate is what happens when you have a head injury. And broadly speaking, whatever the cause is, whether you were in a car and involved in a crash that causes your head to swing back and forth in a kind of whiplash fashion, or whether you are falling from a height onto a floor, what happens is because of the momentum of your head and brain, there is a movement of your brain within your skull. So even if you have a fall and there is no fracture of your actual skull, there will be a movement, what's called a coup and contra coup injury to your brain, where your brain is basically shaken around in your skull. What that means is that if you sustain an injury as a result of a fall, or as I said, a road traffic accident, you're not just going to have one part of your brain injured. So again, this adds to the complexity of the types of problems people may develop after the head injury, but also obviously how those could lead into increased risk for dementia. So what does a head injury do? Well, as I said, the location can be anywhere in the brain, depending obviously on the cause of your injury. But as you could expect, frontal lobe, so the front part of the brain being damaged, is by far the commonest. That tends to be the way we fall, or the area that's most affected in road traffic accidents. And in terms of what we see, if we see somebody in A&E and we do a scan, we may see an element of bleeding or bruising or tissue deformity of the brain. But we are starting to understand that a huge amount of head injury changes or things that happen in the brain after head injuries are invisible. And actually, as I mentioned in this slide, maybe half to 2/3 of people will have a normal brain scan after a head injury, particularly mild head injuries. And what we're starting to understand is that some of the damage that we don't visualize in routine scans is actually there, and again could be a contributor to long-term risk for dementia. And that the particular thing that's been focused on is the degree of axonal damage that head injuries cause. And this cartoon demonstrates what that means. So axons are the tail ends, if you like, of nerve cells, the projections of the brain cells that do the hard work in terms of our thinking capacities. And what happens when your brain is shaken within the skull, as you can imagine, is that those tail ends of cells will get stretched and at a certain point may break and split and tear. And that tearing tends to happen at the boundary that occurs between the grey matter of the brain. So that's the nerve cell bodies, as I said, the areas of the brain that look after all of our main thinking capabilities, and the white matter, which are the tracts between them, both equally important, but that boundary between the two is what's really vulnerable in a head injury. And this shearing, this tearing and stretching of the axons is the thing that currently we don't visualize in routine brain scans. So it's really hard at the moment to determine when people turn up to A&E, have they suffered a degree of what we would call diffuse axonal injury. These two pictures demonstrate pictures of somebody's brain after death. The reason I put them up is because the little black dots on both of these pictures represent what are called traumatic microbleeds. So these are very small areas of bleeding as a result of some kind of trauma, some kind of head injury. And so in the past, we thought these represented the damage that you can't always easily visualize on a scan in A&E. But we're starting to understand that even these small areas of bleeding may not correlate well with the degree of axonal damage, this underlying stretching and tearing of axons that serve these tracts between areas of the brain. And this slide, what this demonstrates is a new type of imaging that's become common, certainly in research, which is called diffusion tensor imaging. And what this does is look at the orientation of water in the brain. And it allows us to visualize these lovely tracts. So this picture demonstrates a kind of green set of tracts that you can see. And it allows us to see where there may be breaks or changes in the structure of these tracts. So for the first time, we do now have a form of imaging that hopefully gives us some idea about this invisible damage that we haven't previously seen. The issue with this is the fact that this type of imaging is really only done in the research domain, because we still don't really understand how we interpret that in terms of clinical symptoms and prognosis after things like head injury. So at the moment, if you have any form of head injury, you will still have what we call a structural scan, either an MRI scan or a CT scan. You would not normally have a DTI scan like this. So coming on to the main reason that I'm here to talk to you, which is about the links between head injury and dementia. Well, to be honest, as Simon Lovestone said earlier on, we know there is a link between head injury and dementia, but exactly how that link operates is still a bit unclear. The fact that we know that people develop cognitive problems with their thinking after head injuries, we've known this for a long time. So there have been cases going back 50 years or more, particularly in people, for instance, former boxers who will often experience multiple head injuries, repeated head injuries. So we know there's an issue. What we're not sure about is whether it is a direct result of a head injury or whether this leads into a subsequent pathology that increases your risk of dementia or not, and what that looks like. More recently, we know that people with more moderate and severe head injuries, so these would be where there is a penetration of the skull, people lose consciousness, for instance, so these are the more severe injuries. The majority of those will report some kind of long-term thinking problems. And one analysis, what's called a meta-analysis, so this is trying to pull all the literature together, looking at all the papers that are out there, looking at this type of theme, in 1991 found that there was about a 1.8 increased risk for developing Alzheimer's dementia after a head injury. And in fact, when you look at those papers, it seemed that TBI was probably the most important factor, only after things like age and family history and what's called the ApoE genotype. So this is a genotype we know that is particularly linked to increased risk for developing Alzheimer's disease and dementia. But there did seem to be some interaction. So for instance, we know if you have this ApoE genotype and you have a head injury, your outcome may well be worse. It's also feasible that if you have repeated head injuries, or if you're younger when you have your head injury, that may again lead to a worse prognosis in terms of developing dementia long term. But this is a long process. So there's been a lot of studies recently done, particularly with this new form of imaging that I mentioned, the DTI imaging, that have demonstrated you can see changes in the brain decades after somebody has had their initial head injury. So you see levels of inflammation, and you see things like higher amyloid levels. And again, Simon mentioned that amyloid is one of the proteins that we see that's increased in Alzheimer's dementia. So we certainly know there are links going on, and we know that, as I said, consequences of head injury will continue for decades after. So at the moment, in terms of what we know, we estimate there's about a 1.6 increased risk for developing Alzheimer's dementia after a head injury, more commonly in men than women. Again, I'll come on to that a little bit later. And so that could equate to about 5% of all dementia cases. So it's not a small factor in terms of what causes people's risk for dementia. This is a very busy slide, and I'm not expecting everybody to read all of this or go through the details. But what this emphasizes is that head injury is just one factor that feeds into our risks for developing dementia or not. So again, as Simon and Ivan have mentioned, we know Alzheimer's disease starts several decades before anybody develops a clinical syndrome of dementia. And there are various risk factors that increase the risk of that disease progressing into dementia. And similarly, there are protective factors. So we know things like if you exercise regularly, if you stay active in terms of your occupation and education, all of these things protect against that progression. But TBI is certainly one of those risk factors that seems to interact with all these other things in terms of increasing your risk for dementia or not. But again, coming back to what I said at the start, it's still not clear. So on this slide, I've quoted a couple of other more recent studies which involved really large populations and looked at the data around whether head injury leads on to risk of Alzheimer's dementia. And actually both of them found no particular risk. And the reason these studies were important is because one of the issues around looking at TBI as a risk factor is the fact whether we are identifying everybody. So there's a problem sometimes with how we categorize TBI, how we identify it, who presents to doctors and A&E and so on. So it's not a clear picture. And just to touch briefly on other types of dementia, things as Simon mentioned, frontotemporal dementia, Lewy body dementia and so on, the evidence is still very unclear. The one thing I do need to mention as we are talking about head injury is this thing called chronic traumatic encephalopathy, or CTE. And this was the first type of pathological change that was described back in the 1920s as a result of repeated head injury. It used to be called punch-drunk syndrome or dementia pugilistica. What's interesting is that it was understood more recently that you see a lot of these tangles, the tau tangles that Simon referred to, in CTE as well as in Alzheimer's disease and dementia. So there's still a lot of debate. We don't really know whether head injury activates some kind of form of neurodegenerative pathway that leads to dementia, whether it accelerates other conditions, or whether CTE actually expands to other areas in the brain as time progresses, and that's the cause of an underlying dementia. So exactly how the changes that occur subsequent to a head injury, whether that feeds into this thing called CTE, and how that feeds into dementia is still very, very unclear. And this slide just demonstrates the similarities we see when we look at imaging of people with head injury and dementia. So this is a slide that shows people who have had imaging looking at tau in the brain. And you can see clearly there from left to right, we've got some control subjects, patients with Alzheimer's dementia, football players, so this is American football players who would be highly exposed to repeated head injuries, and people in the military, again, another very at-risk population. And what you can see is there's a lot of similarity there in terms of quantity of tau in the brain. So there's definitely some overlap, but as I said, still quite unclear how the two things are connected. I'm aware that time is ticking, so I may slide through a couple of these slides.
Yeah, Vanessa, if I could, because we're sort of just gone over the 15 minutes, would you like to sort of open to question after a slide or two?
Absolutely. Yep. I mean, I think the main thing I want to emphasize, and I just want to say something very briefly about the alcohol component as well, as I said, is that how repeated head injuries or even single head injuries feed into risk for dementia is still very unclear, how that sits with this concept of CTE or not. And I guess, as I said, I apologize that I've ended up over speaking to the slides. The one thing, I'm just going to move on very quickly to say something very briefly about alcohol and TBI. And what I chose to do was to focus on if you drink on top of having a head injury, what kind of result that may lead to. And so I am just going to slide through my slides very quickly. I apologize for that. The main problem we have is that we do know that obviously if you have an existing alcohol use disorder, so those are people who are dependent on alcohol or misuse alcohol, you are more likely to have a head injury. That's probably not a surprise. But equally, if you sustain a head injury, you are more likely to start to drink or misuse alcohol afterwards. And so the two can feed into each other and certainly lead to a worse outcome overall. The other problem is, as you can imagine, that people who have alcohol misuse problems tend to also have a range of cognitive issues as well. And so that can cloud the presentation of head injury and cloud what is causing those problems. And on top of that, we also know that alcohol can cause alcohol-related brain damage in its own right. It may be secondary to those head injuries that I mentioned, to thiamine deficiency, or to the direct effect of alcohol on nerve cells. So the issue of how alcohol interplays with head injury is very, very complex, not an easy one to tease out at all. So to summarize, as I said, tried to bring this together to a prompt end, and I apologize for the fact I've not been able to cover everything I hoped. The bottom line is that TBI, head injury, is very common. The outcome is very varied and difficult to predict. We know that there is some link between head injury and forms of dementia, but what that pathway looks like is still very unclear. We know that people who have alcohol use disorders, both before and after having had a head injury, leads to poorer outcome. We know that we really need to have more public health interventions to address this. So one of the slides I didn't get to mention talked about the fact that public health initiatives like wearing seatbelts in cars, reducing the legal blood alcohol limit has had a massive effect on the amount of head injuries that people have had. So those kind of interventions have had real change. But what is obvious is that we still lack a lot of clinical research around head injury and how alcohol impacts on those risks. And to be frank, as a clinician, one thing I've certainly found over the years is that services do not support people well who have head injuries by and large. As I said, most of them do not present to A&E, do not go and see their GP, and yet they may continue or develop new difficulties years after. So clinical services, as clinicians, we need to think about how to adapt those, particularly if, as Simon said, we're at the point where we could be talking about preventing dementia, or at least developing treatments that could be used in that preclinical group before people actually develop the dementia syndrome. Then TBI becomes a very important thing that we need to be able to identify and ideally try and prevent where we can. So we reduce those risk factors and we can optimize the likelihood of response to treatments. As I said, again, I'm very sorry I wasn't able to cover everything, but this is a complicated problem. This last slide just emphasizes that. It's not a clear answer to this one, of course, but I hope that we can make some progress over the coming years.
Thank you very much, Vanessa. That is a great talk, and that's very kind. So you've left your email there in case anyone has any other questions. The first one, I guess, goes back to something that Karen asked, whether you're aware of any studies looking into the long-term outcomes of ECT, electroconvulsive therapy, in regards to dementia risk.
So, I mean, broadly speaking, the research that is out there suggests that there's no specific link with ECT and dementia, to my knowledge. I think you have to think often why somebody may be requiring ECT for treatment, and obviously those tend to be people who have very often very complicated presentations and may have a whole range of other problems going on, and that could include some kind of underlying problems within the brain as well. One of the points that I wasn't able to get to in my slides was the fact that one of the problems about teasing out the impact of head injuries in the progression of dementia or not is because when someone has a head injury, we don't really know whether that person already has some kind of level of underlying Alzheimer's disease or not. Unless that person has already been seen clinically or had a brain scan or something that can give us some idea, it's very hard to tell. So if over the following years they start to progress in terms of having more memory problems, it's really hard to know, is that because of the head injury or is that because they had an underlying dementia that was developing anyway? And I think the same goes for ECT. My understanding is there's not strong evidence as a direct link, but as ECT is often, for instance, used in the elderly who are very depressed, it may be that those people already have some underlying process that's contributing to the development of dementia, whether or not they had ECT. So I think that's the kind of problem with that question. It's a very good question, but I think it's very difficult to tease out what could be causing problems.
That's great. And Michael Ben asked another question that's specific to brain injury. He's wondering, can it be treated to minimize negative outcomes, or is prevention the only way to avoid its consequences? Is there anything that can be done in the acute or the post-acute phases to minimize the risk?
Yeah, so currently in terms of any traumatic brain injury, there are no specific treatments. There are various treatments that have been used pharmaceutically, but these are all symptomatic treatments. So this is about, for instance, if somebody's having problems with their attention or their memory as a result of their head injury, there are certain medications that can help with that. Depression is a really common problem after head injury, and obviously that could affect your ability to concentrate or remember things. So antidepressants are used very commonly after depression. And there's some research going on as to whether things like giving antidepressants before people develop depression after head injury may be useful. And then there's various behavioral and psychological therapies that are used post-traumatic brain injury. But unfortunately, there's no pathological treatment, if you like, that at the moment we know will change any of those progressive changes over the years. The biggest results to date have been about trying to prevent injuries occurring in the first place. So the public health initiatives around alcohol, driving risk, that sort of thing. Ivan, I'm not sure if we've lost you for a moment there. Your screen has frozen.
Hi, Vanessa. It may be that Ivan is just, as you say, locked for a moment. Having a look at the questions on YouTube, I think we've covered most of them. And as we've mentioned, we'll put any unanswered questions on the website, and we'll share a link via the Eventbrite invitation. I suggest perhaps now we can go to Professor John O'Brien, who is our next speaker, and hopefully we should have Ivan back with us in just a moment.
Thank you, Heather. If I could just say one thing as well, I'm not sure if we were planning to try and share some of our slides, but as I didn't have time to go through anything, I'd be more than happy to do that so that people that have attended this seminar can see everything that I'd hoped to present in that time, and apologies again for having to whiz through. Thank you.
Thank you for that offer, Vanessa. We've actually had that request come through already, so you've answered that one, and we've said yes, that we will put speakers' slides in a PDF form on the website. Just give us a couple of days to organise that, so hopefully at the beginning of next week that will all be available. So thank you very much, and now we go to Professor John O'Brien. Thank you.
Hello, well, thanks Ivan and others for the opportunity to be part of this Great Minds, Great event. And my topic is brain imaging for dementia. And in common with many of the other speakers, I not only have research interests in what I'm talking about, but I also run clinics and see people clinically. And one of the excitements for me in brain imaging is having seen over the years how imaging has moved from something that we can use to investigate underlying biology, if you like, into the clinic, where we're actually using it to better diagnose and manage people with dementia. And if we look at this slide starting at the bottom, we can use imaging to look at underlying causes and mechanisms, a bit like Simon was talking about in terms of what are the actual basic things that are going wrong that we might be able to put right. We can use it in clinical trials, both to select people who are suitable for certain trials and also to use it as a marker for outcome in studies. But we can also use it very much in the clinic, not only to rule out other brain disorders, but to assist with subtype diagnosis, by which I mean dementia is obviously just an umbrella term. And we really want to find out, do people have Alzheimer's disease? Do they have vascular dementia? Do they have frontotemporal dementia? Or do they have Lewy body dementia, which are the big four, or indeed sort of something else? And my talk is really going to be some examples from each of these different ways that we use brain imaging. We think of brain imaging in two main types. One is structure. That's looking at what the brain actually looks like structurally. And the two modalities here are CT or computed tomography or magnetic resonance imaging. And many people might have had one of these scans. They're not only used for scanning the head, they're used for scanning other parts of the body as well. But we're not only interested in what the brain looks like, we're interested in its function as well. And there we have a number of different ways of looking at function. We can look at blood flow, we can look at energy, how the brain loses energy. We can look at specific chemicals in the brain, and we can look at specific proteins, and Simon showed an example of looking at amyloid. And we can also look at other processes, and the ones in red here are really only research uses at the current time, but we can look at tau, we can look at inflammation, look at synapses, which again are both processes of real interest, as Simon illustrated in his talk. So in terms of the clinic, when we have people presenting, we might do a scan, in this case a CT scan. This is someone who presented to a memory clinic I was working in, and you probably don't need the arrow to see that there's something there that shouldn't be there, a large lesion at the front of the brain, underneath which is quite a lot of sort of blackness and darkness, and that's brain swelling or edema. This was someone who presented with a gradual onset of cognitive problems, and the provisional diagnosis before the scan was early Alzheimer's disease. And in this case, it was a frontal meningioma. Fortunately, that's a benign, not a malignant lesion, so that could be removed to help the symptoms. And so clearly in this case, it was to exclude a sort of non-dementia cause for the cognitive impairment. And that's still an important use of imaging, although admittedly, we don't pick up many tumours, but there are some people and they are there. If we look at a CT scan now of someone with Alzheimer's disease compared to a healthy control, you can probably see that the person with Alzheimer's has slightly more in the way of blackness there. That's fluid, cerebrospinal fluid. And this shows sort of generalized brain shrinkage or atrophy. But the problem with this is that we see this in a number of different disorders, not only dementias, but it can occur as a part of the aging process. And so some of these changes are not so specific. But there are other changes on imaging, and now we're looking at a magnetic resonance imaging scan. The arrows show a medial, the inner part of the brain, part of what we call the temporal lobe and the hippocampus, and this is a part of the brain very much involved with memory and new learning. And this is one of the areas we know from a number of studies is affected quite severely and quite early in the process of Alzheimer's disease. So by doing a sort of MRI scan and focusing in on this medial temporal lobe, we can start to see changes that are now characteristic of different types of dementia, in this case Alzheimer's disease, as opposed to the subject on the left, it's a healthy control for comparison. And what we see is this kind of change in about 80% of people with Alzheimer's and about 10 to 15% of control subjects. So what this immediately tells you is that while this is a helpful change, it would point us towards thinking that Alzheimer's might be the diagnosis. It's certainly not 100% accurate. And this is the same with many scans I'm going to show you today, that they don't have absolute diagnostic accuracy. So we're not going to make a diagnosis of a type of dementia from a brain scan. It's going to be supportive information, and it's going to add to the clinical picture and the cognitive testing. And that's very much how we're using imaging in the clinic. Now that's looking at a scan at one point in time. What's particularly helpful, especially for research studies, is scanning the same person twice. because all our brains are different shapes and different sizes, and one person's scan can look very different from another. But if we take the same individual, the same brain, and we scan the person again at a later date, maybe 6 or 12 months later, we can directly compare the scans, and we can look at things like whole brain volume loss, which is illustrated in this slide here. And we can see that it differs between people with Alzheimer's disease, who have a brain volume loss of about 2% per year, compared to control subjects, or indeed some other types of dementia, where the volume loss is much less, maybe about 0.5% per year. And the importance of this is not so much diagnostically, although it can be very helpful in a clinical setting. If someone has memory problems, to do a scan and look back if they had one maybe 3 or 4 years ago to see if there's been a change. But this can be used as an outcome measure in clinical studies, where you have groups of people who are on a drug that you hope will slow the degree of brain shrinkage, and a group of people who are on a control or a placebo. And you can actually compare the rates of shrinkage between groups to see whether the treatment seems to be helpful or not. Now, most of the imaging that we do at a clinical basis is done on clinical scanners at certain magnetic strength. It's called Tesla, 1.5 Tesla. 1 Tesla is about 30,000 times the magnetic field of the Earth, to give you some idea. And you can get, as you see at the top here, quite a nice picture of the hippocampus and this medial temporal lobe we were looking at earlier. But if you increase the field strength, this is going up to 3, we can now go up to 7 and even higher, you get better resolution of the brain and of different parts of the brain. And what this higher field strength shows here is that we're not just identifying the brain structure, the hippocampus, we're actually able to see in some reasonable detail the individual subcomponents of the hippocampus. And this is, to me, very exciting, because this is the sort of detail that previously you could only see under the microscope. You could never see this in living people with dementia. And so we can now start to see that and see some of the early changes and ask whether some of these changes are helpful, for example, with regard to early diagnosis. In vascular dementia, which has also been mentioned, we can use imaging, and magnetic resonance imaging is actually the modality that's particularly helpful for showing vascular change. And we can see some of the changes that are illustrated here. Strokes or infarcts can be well seen. Also more diffuse vascular white matter change, as can be seen in the bottom right here, and also some microbleeds and microinfarcts. So we can start to see vascular changes and tease out whether someone might have a vascular contribution for their cognitive impairment or their dementia. So these findings have led NICE, National Institute for Health and Care Excellence, to suggest that they're so robust and clinically useful that we should be doing these within the NHS, and that's really very important. And so in their guidelines in 2018, they said that structural imaging, so that's CT or MR, should be offered to people in secondary care if they're coming along for specialist memory or dementia diagnostic assessments, to both rule out reversible causes and assist with subtype diagnosis. Note this is offer, because obviously people might not want to have a brain scan and some people don't, but there are good reasons why they can be useful. And then the second recommendation was that if the dementia type wasn't clear and vascular dementia was suspected, that MRI was better than CT for demonstrating that. So that's really what structural imaging can offer. If we move now to functional imaging, this is also something that has reached the clinic and is NICE recommended. This is now looking at glucose or metabolic imaging. So this is how well the brain is functioning, how much energy it's using at rest in the scanner. And on the bottom left here, you can see a control subject, and areas of yellow and white are areas of quite high metabolism. And this is a very normal looking brain. Most of the metabolism takes place in the grey matter rather than the white matter, but you see quite good areas of metabolism. And you can contrast that with the subject with Alzheimer's disease, who has quite marked loss of metabolism in many brain regions, particularly in temporal lobes, parietal lobes, and some of the frontal areas as well. And this can be helpful if after a clinical assessment and a structural imaging, it's not clear whether the person has a problem or what problem they have, then often a metabolic scan like this PET scan can be helpful in helping to support the diagnosis of Alzheimer's disease. And similarly, using looking at metabolism, or in this case, another technique, which is blood flow, again, using functional imaging, we see here in frontotemporal dementia that people's frontal lobes can be demonstrated to be lacking in perfusion, and similarly they will be lacking in metabolism. And again, NICE has suggested that in the clinic this can be useful in terms of assisting with the accurate diagnosis. And other dementias might need other forms of scanning. So here we have a more specific form of scanning where we have a particular ligand or a chemical that's injected into people that gets into the brain and highlights the dopamine system, in this case, a transporter for dopamine within the brain. And you see a very different sort of scan, but this is basically showing the level of dopaminergic activity and integrity in the brain. And in the left-hand side, there's a normal subject, and that is the normal appearance. Dopamine is very much concentrated in deep structures in the brain called the basal ganglia that are responsible for movement. Is someone with dementia with Lewy bodies, which is a type of dementia that is akin to the dementia that develops as part of Parkinson's disease. So people have often motor problems, but they also have dementia, and they may have hallucinations and fluctuation. And here you can see that the scan looks very different. There's a loss of the dopamine system. And actually in dementia of the Lewy bodies, brain structure is often relatively normal, so it can be hard to use that in terms of a diagnosis. But the loss of dopamine is very useful, and again, abnormal in about 80% of people. And again, a nice recommended tool for clinical diagnosis. Then we move on to amyloid imaging, and we've seen examples of this, and it was one of the holy grails in a way of imaging to try and get to image some of the protein abnormalities that we see in Alzheimer's, whether that's amyloid or tau. And around about 10, 15 years ago, the ability to do this came along. We actually had several companies develop ligands for amyloid, all of which seem quite good at demonstrating brain amyloid. And they're illustrated here in the top is the three people who just have binding in the white matter, which is a relatively normal, non-specific finding. And then in the bottom, we have examples from 3 different ligands showing increased uptake, particularly in the grey matter, indicative of large amounts of amyloid or plaque pathology there, highly suggestive of Alzheimer's disease. Is seen in some other disorders, but very supportive of Alzheimer's disease. Now, this is available for clinical use, although it's not used very widely at the moment, but it has been approved. Maybe it's more common usage, though, is still in terms of research, and we heard before about the long sort of prodrome, or how long Alzheimer's disease takes to develop. And this was partly shown through studies like this from a group in Melbourne who did some repeat imaging, but also some modelling, and demonstrated how amyloid levels would be projected to increase over time from a stage of sort of normality at the bottom left here to a stage at which Alzheimer's disease is diagnosed. And the length of time is very, very striking. You can see here maybe 20, 30 years between moving from a stage of normal amyloid to one where Alzheimer's disease is diagnosed. And that allows an early prodrome for identification of people for early studies, but also shows we've got a very wide window where intervention might be possible, whether that's by a drug or by some change in behavior. And things like this have really transformed, I think, our understanding of the disease process and how and when we might want to intervene. Now, Simon hinted at this before, and this is just a sort of extreme example of some of the issues with some of the early studies that were done on amyloid immunotherapy in Alzheimer's disease. These were done on the basis of asking people to take part on a clinical diagnosis of Alzheimer's disease, without actually having the ability at the time to really do brain scans to see if they had evidence of increased amyloid in the brain that would suggest they might be people who might benefit from a sort of amyloid lowering agent. And some of these studies, in this case, in the sort of less genetic form of Alzheimer's, if you like, it transpired that over a third of people entering the study actually had normal scans. So didn't actually have the pathology that the treatment was aiming to reduce. And it's partly because of that it's very standard now in current studies of amyloid lowering agents that a requirement is there at entry for the demonstration that there is amyloid either on imaging or on spinal fluid. And that's an example of subject sort of stratification or selection for studies to make sure the right sort of people are coming into the studies. Here's an example of the use of repeat amyloid imaging. This is at baseline on the left-hand column in 1 year in an antibody study, which there's been quite a lot of interest in, aducanumab, which there seems to be a signal, certainly signal in phase 2 and in some of the phase 3 studies. It's an antibody which is designed, like many of the others, to lower amyloid levels in the brain. And you can see here from the scans, I think that if you compare the people who had different doses of the antibody infusion compared to placebo at the top, you can clearly see that the brain imaging shows that the treatments seem to be quite effective actually in reducing the amyloid burden. And that's one way brain imaging can be used as sort of outcome measure to show that the drug is actually identifying the right target in the brain and actually doing what one hopes it will do, which is actually lower the drug. And then, of course, one hopes there's a clinical response that mirrors that, which is often where the debate is about as to how good that clinical response actually is to mirror the change in the biology. We've, in the last few years, had the ability to look at tau as well, which is very exciting. There's several agents that can do that. Tau is the constituent of tangles, which has always been quite difficult to image because these are often intracellular and hard to access. And this is just an example of a study we've done in Cambridge, which I won't go through the details, but it's using a particular tau ligand using PET scanning. So again, it's a functional scan. So people are injected with this ligand, and then we do a PET scan. And then we can see areas of high uptake here shown in red and green. And we can show in the people with Alzheimer's disease that they have quite high levels of binding in parietal areas, in temporal areas, and also deeper in the brain compared to healthy control subjects in the bottom. And in this study, we had another group of people with another disorder. It's called progressive supranuclear palsy. But the point of it is that it's also caused by abnormalities in tau. But in this case, the tau is not in the cortex, like in Alzheimer's. It's very deep in these deep structures, the basal ganglia. And we were able to show using this chemical imaging method for tau that clearly, although both Alzheimer's and PSP are tauopathies, they have very different distributions of tau, very much in keeping with what we would expect based on the pathology of the two disorders and what's known about those. So that's a bit of a whistle-stop tour of imaging, but just to summarize, it is in clinical use and it's being used in memory clinics up and down the land to help diagnose all the common types of dementia. With regard to research and moving forwards in finding therapies, it's increasingly being used as a marker for selecting people for trials and as an outcome measure. It's worth noting that the UK, we're fortunate to have some of the best imaging in the world for dementia through the Dementia's Platform UK. We have a multi-site PET-MR network, so these are state-of-the-art scanners where you can do PET imaging, functional imaging, and also MRI scanning at the same time. And we also have a UK 7T MR network. So this is high field strength MRI scanners where we can get that high degree of resolution we were looking at earlier, which is potentially very valuable for studying people with and at risk of dementia. And I think imaging has and will continue to have a central role in many research studies, both in those with and at risk of developing dementia. Thanks very much.
Thank you very much, John. Fantastic presentation. We have about 5 minutes for questions. I guess one question just coming in is, going forward, what do you hope imaging will be able to tell us that it cannot tell us at the moment? And what sort of innovations do you hope to see in the future?
Well, I think ideally we'd use it for more individual patient management, which we're not really at the moment. We're using it as a broad diagnosis, and we would like imaging that was slightly higher in diagnostic accuracy. I think that is starting to come with some of the amyloid and the tau agents. But I think what I would really like it to do is to be able to personalize it to individuals, to say, I've done this scan, you have a lot of this type of pathology, I think you'll benefit most from treatment A. So Simon talked very eloquently about not just one treatment being given. I think that's incredibly important. So we can fairly easily pick up on the scan the contribution of vascular pathology that's likely to be contributing. It would be nice to be able to use that also in other ways, tau, and as many of us increasingly think other avenues such as targeting brain inflammation might prove useful. If that is going to be the case, again, how are we going to work out who are the right people to target some of those therapies to? And I think imaging has a role, at least initially. I don't think we're going to be in the clinic asking people to have 4 types of brain scan and giving them treatments. We'll want better markers, for example, in blood eventually to do that, but it'll be an important translational step in working that out.
And on the point of innovation, you talked about PET tau, and as you know, something that's very typical about tau in Alzheimer's disease is that it follows this almost like a predefined path. And so there have been some studies already asking the question of whether you can stage Alzheimer's disease using this technology. So what are your thoughts on staging Alzheimer's disease using these technologies? What's the role?
I think that's an important part of what we're able to start to do now. We still need a little bit more validation, I think, in that, and particularly the always quite challenging separation of apparently healthy older people who do have some tau that starts to overlap with the tau that you see in people with Alzheimer's disease. But I think starting at that kind of early stage before you get widespread brain involvement is likely to be very important. We're finding out more about the association between tau and atrophy, the brain shrinkage. And it seems to me that by the time you get shrinkage of the brain at a sort of global level, it's going to be very hard for treatments to really tackle that. But if we can start to pick up early changes in tau before we get that atrophy, which we think does happen first, the tau before the atrophy, then that will be the time to actually find people and be able to help them with therapeutics, which will hopefully be developed over the next few years, as we heard.
Yeah, and I guess one last question with your clinical hat on, which is coming from Susan Finch about the value of currently available medication for dementia. You know, she's giving the example of donepezil being delisted by France due to lack of medical evidence. What is your view on the sort of tools that we have available to us at the moment?
Yeah, well, I think they're limited, but I do think they're effective, and it's puzzling that we wouldn't want to use effective and fairly safe treatments for people with Alzheimer's disease and indeed Lewy body dementia, where the treatments also seem to work. They're modest benefits, but the clinical trials show without any shadow of a doubt, I think, that there is benefit. There were discussions earlier when these drugs were very expensive about cost effectiveness. They're now very cheap. They've come off patent, they're generic. So those arguments don't really hold sway. And NICE in its recent economic modelling, well, it's a couple of years ago now, they clearly showed they were cost effective for the NHS. And indeed, not just one therapy, but that it was probably best, certainly for people who fit the requirements, to be not just on one class of drug, cholinesterase inhibitors like donepezil, which are the most widely known, but actually to be on dual therapy as another drug that acts on the glutamatergic system, memantine, and that actually the benefit of both seems to be additive. So it would be terrible, I think, to deny people the current treatments, even though we acknowledge they're just a first relatively small step to where we want to get to.
Yeah, thank you very much, John. And I think it's fair to say that we don't necessarily yet understand why it tends to work better for some people, while for others. And this goes back to the question of personalized medicine. And I think that there will be a role to be played there. Okay, fantastic. Thanks very much for sparing the time and for your thoughts and the talk. We're moving swiftly on to Dr. Setelu, who is a researcher at Cardiff with the HealthWise Wales cohort, and she's going to talk about the burden of dementia, of caring for patients with dementia. Over to you, Fatani.
Yep, just sharing my screen. Okay, I think it's good. Okay. So yeah, as you mentioned, I'm a research associate working with HealthWise Wales in Cardiff University, and today I wanted to talk about care burden in dementia and how it fits with what we do in HealthWise Wales. First, I wanted to give a brief background on what being a carer means. So when we're talking about informal carers in research, we mean a person who provides some type of unpaid ongoing assistance with activities of daily living to a person, including family members, friends, neighbors, who have a chronic illness or disability. This definition does not include those who provide paid care as part of formal occupations, such as nurses. So we know that informal caregiving is an integral part of many societies, and it's linked to an aging population and increasing reliance on unpaid carers. The care-related responsibilities may vary according to the care recipient's condition and needs, which could range from a mobility difficulty to chronic mental health problems or dementia. And again, from the extent of the carer's contribution, ranging from a few hours per week, such as helping with shopping, to full-time daily caring responsibilities, including dressing and washing. So we know that caring for a loved one can be a demanding role, with lots of responsibilities increasing and disrupting one's own daily routines. Carers may experience distress and poor mental health due to their caring duties, but can often remain hidden from services that could support them. Looking after another adult or family member can take up a lot of their time and result in neglecting their own well-being. So there is an increasing concern about well-being of carers and how they manage those responsibilities in conjunction with their physical and mental health. We know that stress among carers of patients with dementia is also linked to patients' health decline and poor quality of life. And in particular, we know that patients' physical dependency has been linked to care depression, while also behavioral disorders like disorientation can be very crucial in the experience. These symptoms could lead to transition to institutional care and have been shown to cause greater burden than cognitive or even functional impairment in some cases. So overall, we know that care burden is a complex phenomenon and linked to both the framework of care and potentially leading to negative consequences for those involved in the caring process. Carers of dementia, patients with dementia, suffer disproportionately from severe stress due to coping with behavioral problems of the care recipient, potentially inability to communicate, feelings of intense loneliness or even loss. This can lead to disruptions in the carer's life and subsequently to physical and mental health problems, especially when the carer has trouble coping with the caring situation. This situation can put the carer at an increased risk for anxiety and have a negative effect on the carer's health. So poor mental health can compromise the carer's abilities to maintain themselves adequately in their caring role. So care burden can also be aggravated in cases that the care recipient refuses to accept assistance from other potential carers, as they might not see the need to do so, or because they might not want to receive help from outsiders and only accept help from a family member or a close one. And the care recipient's problematic behavior, along with the severity of condition and impairment, either cognitive or functional, can add further to the stressful situation of caring. The framework of care is also important, as the lack of support and the presence of dysfunctional social interactions can have a negative impact on the carer's experience. So all these issues can contribute to the management of care provided, resulting in poor standards of care, as patients with dementia are, as we know, are heavy users of health services with very complex needs. So how does HealthWise Wales fit with what we know about care burden and in general and in terms of dementia patients? I just want to describe you HealthWise Wales to give you a context. It is Wales' national web-based longitudinal health study, which is funded by Health and Care Research Wales. It is a database of 40,000 individuals who live in Wales or access their healthcare in Wales and are aged over 16 years. This includes everyone, even students who may only be temporary residents in Wales. HealthWise Wales is open to everyone, and those who register to consent to be recontacted about participating in other studies. Though no questionnaire is compulsory, the strength of HealthWise Wales is that it collects data from that are not routinely available from other sources, but also it can facilitate collaborative research with researchers within external to the health and care research infrastructure through identification of potential participants, collecting specific data on a range of topics, including individuals' opinions on medicine wastage or caring and prescribed medicines. These types of questionnaires are designed and developed in collaboration with researchers who have expertise on the relevant field. So other than those questionnaires, external questionnaires can be developed through collaborations with third partners, such as the link that was established with the Dementia Platform UK. So we identified the Great Minds project as a way of delivering dementia research opportunities to our participants. All our participants aged over 40 can volunteer to take part in dementia studies by signing up to Great Minds and completing online cognitive tests via the HealthWise Wales platform. So why is HealthWise Wales working with Dementia's platform? Many HealthWise Wales participants and their families have been affected by dementia. So our participants have suggested dementia as a priority HealthWise Wales research focus. We know that the causes of dementia start decades before symptoms show. So the best people to help study these causes are healthy volunteers. So the link between lifestyle questionnaires and other HealthWise Wales data to the cognitive questionnaires from the Great Mind project will allow us to make connections between lifestyle and environmental factors and brain function. So these types of research initiatives can help to understand and predict dementia progression and impact on the patient and the family. So at the same time, our research on carers has focused on self-reported data that include questions on care and caregiving responsibilities. These questions are available through the core modules I mentioned before, and we have used them to explore the needs of carers within HealthWise Wales. The first is a way of identifying the level of caring responsibilities per week. And the second explores the potential impact of a caring role on employment. of the carer. So these are some of the key research findings from a study we conducted focusing on carers' mental health and health behaviors. So we can see that more carers experience common mental health conditions than non-carers. We also explore the level of caring, though this is not presented here for brevity. Carers who have given up work to care for someone report poor mental health compared to other carers. These findings suggest that caring can negatively affect a carer's life, especially their mental health, highlighting the need to better support their mental health needs. Carers generally reported more unhealthy lifestyles behaviors than non-carers, including reduced physical activity, unhealthy eating, and smoking. Carers with more of these unhealthy behaviors also had worse mental health. This could be explained by the fact that the time and energy a carer devotes to daily caring activities could lead to this neglect of their personal health. So I previously described a collaboration between HealthWise Wales and Great Minds as a way of recruiting participants for external questionnaires. However, this doesn't include only questionnaires and cognitive tests, but also different types of research, such as in-depth interviews. And one such collaboration was developed between HealthWise Wales and David Wasley, a researcher from Cardiff Metropolitan University, who aimed to investigate the experiences of long-term voluntary carers of people with mental illness. And in relation to their caring demand, the social support they receive, their coping mechanisms, and the link to psychological well-being. So HealthWise Wales carers received an invitation to participate to the study. Those expressing an interest were put in contact with the researcher, and the study was in two parts: a short preliminary telephone call to answer some initial questions, and a longer telephone conversation to learn about their experiences as voluntary carers. So from this collaboration, 10 interviews were completed, and analysis of the interview data is underway. Another interesting project focusing on carers will use an arts-based methodology to engage young people. We know that, as I mentioned, caring for a family member or a friend can have negative effects on health and social activities, especially in the case of young people. So young carers may have difficulty voicing their needs, while music has been shown as a creative outlet for engaging with and acting upon research findings. So for this project, we'll be collaborating with Ministry of Life, a community interest company who works with marginalized young people to create music outputs and other creative outputs. We aim to help young people and young carers in particular to communicate important health messages. We're also planning to share those outputs among carers, participants, and other stakeholders through social media to raise awareness and improve knowledge on what it means to take part in research and improve the way research studies are presented to the public. The project has now evolved in a virtual setting due to the COVID environment, and we hope that this transition may help provide some respite during the lockdown and reach out to those people that might have otherwise been missed due to their caring responsibilities and not being able to attend such events. So in terms of our future research steps, additional analysis on care burden by the health needs of the person they care for using the data on caring and prescribed medicine is underway. We also plan to analyze the data collected through the collaboration between Dementia platforms and Great Minds. Also, we aim to share the music video and other outputs from the interactive activity to carers and across our participants. This will also save the way we engage with vulnerable groups such as carers and as well as our participants in general. So while working on a range of projects aiming to identify the health needs of carers, we also offered our participants who have told us that they are involved in caring the opportunity to become our care needs ambassadors and talk about their story as carers. 4 of them have agreed to become our ambassadors, and one of them even provided an interview to our public relations communications partner Yogi Combs about her experience as a carer and how her role has motivated her to be an active participant. So I just want to summarize some other key points about policy. As the previous speakers mentioned, we have a lot of people that have dementia across the world, and the number of adults providing care to their loved ones with dementia is growing. So caring can affect every part of the carer's life, leading to the carer's needs being overlooked. As a carer's ability to cope effectively is linked to the patient's survival and well-being, the impact of dementia should be considered by professionals. Carer's physical and mental health needs should also be considered as part of dementia care. So there is a need to reach carers, especially those who would not consider their distress as a mental health problem and thus not access the needed care that will help them. As primary care is the point of first contact, both in initial detection of dementia and effective ongoing management, targeted interventions such as perhaps education or family support can decrease the burden of carers of patients with dementia. So some final key points. Overall, we know that carers report more unhealthy behaviors and worse mental health than non-carers. This can be worse off for carers of patients with dementia who experience more unmet need for help with different care activities due to an increased need for help with tasks, especially when the carer is the sole support for an elderly person, and they have to reduce their work hours. Lack of family support or even family conflict on treatment, inability to arrange for assistance from other sources such as friends or neighbors. These, along with other issues like negativity or over-involvement in the caring process, may increase care burden and the carer's personal experience of distress. So the burden of caring for a loved one with dementia, and in general as caring for a loved one, should not be studied on an individual basis, but as a complex interplay between the carers and the care recipient's needs. Thank you for listening. And I wanted to leave these acknowledgements for the HealthWise Wales participants that provide their data, the Health and Care Research Wales and SAIL. And I think I'm on time.
Thanks very much for Tani. That's great. I wonder if I can ask you a couple of questions. One is, there is a lot of talk at the moment about the value that digital technologies may offer to assist people caring for those with dementia. What is your view? What do you think are the most promising things as sort of innovations in the field?
I think that as I've seen through the Young Carers project I mentioned is the I stopped sharing the screen, right?
Thank you.
Yeah, okay, sorry. As you can see is that we can now reach people that otherwise would not have been able to attend not only events like what we're doing, but even support for them. And especially in the situation that we're now due to the lockdown, being able to provide support through online means is very useful, and especially for somebody like a carer who might be in a house with their care recipient and not having the ability to do any activities outside the house. So yeah, I think online resources are very useful, especially for carers, but in general for all people that might struggle with their mental health.
Yes, and I guess that there is a question of how the current situation with the lockdown has affected the care of people with dementia. I wonder whether you can share any experiences of your group, of how they're finding the experience.
We know that people already experience disruptions in terms of their daily routines from caring. So putting a lockdown and all the stress about COVID because they're caring for somebody that is already vulnerable with experiencing dementia. So adding all this stress about extra risks about their health and being trapped or having to do activities only inside the house can be very stressful. So yeah, we know that this has put an extra stress on the people that are caring for somebody, for a loved one.
Great. Okay, fantastic. Thank you very much for Tony. And I guess a question to both you and John, just thinking to the future, where do you think the field of dementia should be? What should we be aiming for in, say, 15 to 20 years' time? What is a reasonable goal from your point of view of things to attain, to know that we've succeeded? I don't know who wants to go first.
That's a tough
I've been
question.
talking a lot, so yeah.
That's a big, tough question, Ivan. I think that I would hope we've made better progress with treatments, that we have disease-modifying therapies that are available within the National Health Service. I think it'd be disappointing if we didn't have that. I would like to see us being able to offer people a diagnosis at an earlier stage. It's still a huge problem, as came out with some of the questioning, and we've all experienced this, of people struggling quite a lot to get people to recognise there's a problem in themselves, in their parents, in their brothers or sisters, in their spouses. So I would like us to be more able to offer that, and we're moving in the right direction. And I think there's a lot been done from a societal perspective as well that's been very good, and I think the UK has been taking the lead in some of this with dementia-friendly initiatives, places, use of technology to support. There's a lot more that we can do. So a better use of that in our everyday lives, particularly focused around supporting people with cognitive impairment and dementia.
Great. Any thoughts for Tani on where you'd like to see us in 15 to 20 years' time? What will be a success?
In terms of carers, I think that would be to be able to identify them earlier, especially because there's somebody that attends, for example, GP appointments or other health needs along with the care recipient, but they're usually on the back of that. And I think being able to identify them earlier and provide them the help they need, or even if it's just some information on where they can get help for the patient that they're caring for, that would be, yeah, great as a first step.
Fantastic. Okay, I think we're just coming up to 4 o'clock. I'd like to thank you all. So we've had a fantastic list of speakers. Vanessa and Simon had to drop off due to other commitments, but thank you very much, John and Fatani, for staying all the way to the end. And I'd like to thank everyone else who spared the time to join, and for all your questions. If we didn't manage to get any answer, then we'll post them on the Great Minds website. Thank you all. Thank you for joining us, and I really hope that we'll be able to do this, a version of this again soon. Thank you.
Yeah, thank you.
Thanks. Bye.